Dear learners, I am P. Varadarajan, Assistant Professor working in Sri Kaliswari College. Now we are going to discuss about molecular docking. Molecular docking is one of the most important topic in bioinformatics. So bioinformatics is a field of it is originated from hybrid technology as originated as a hybrid technology from biological science and computer science with the association with mathematics. So mathematics, biological science and computer science all together worked with and the emerged field is bioinformatics. So this is the field for science in which biology, computer science and information technology meet together and we can develop a new field called bioinformatics. With the help of bioinformatics, we can do wonders in biological science by creating a different algorithm. We can solve the structure of our own protein. We can analyze the sequence, both the gene sequence and the genome sequence. We can construct a phylogenetic tree, and we can identify any mutation takes place in the particular gene sequence or not. So we can do wonders with the help of bioinformatics. So molecular bioinformatics, the definition for molecular bioinformatics involves the use of computational tool to discover new information in a complex data set that is from one-dimensional structure to two-dimensional, three-dimensional, and four-dimensional structure. As I have told you earlier, it's a multidisciplinary course. So since it is evolved from computer science, biological science, and mathematical science, we can that is why it is called as multidisciplinary field. We can apply the bioinformatics in molecular biology, genomics, mathematics, statistical techniques, numerical analysis, algorithm, data management, image analysis, evolution, biophysics, biochemistry, and genetics. So where do the data come from? That is one of the important questions. So we can derive data used for bioinformatics from sequence, or we can get data from structure, already predicted structure, or we can get data for bioinformatics from literature or domain or networks. So these are the different ways by which we can derive data for bioinformatics purpose. And the moment you are going to use bioinformatics, it is very important that the databases are very, very important. Because the biological databases are, biological data are stored in the biological databases. The most important biological database is NCBI, DDBJ, and the EMBL. These are three major databases used, frequently used for bioinformatics. And the reason for this important is these three databases are integrated with each other. NCBI is maintained at GenBank. The GenBank is a database maintained in the NCBI. DDBJ, DNA Data Bank of Japan, it is maintained in Japan, and the European Molecular Biology Laboratory. So these databases are integrated. That is why we can get data from any. We can get data from any one of the databases, and also we can store the data in any one of the databases. So the databases are classified into different types. The primary database, secondary database, and the tertiary database. For primary database, these are the example. Nucleic acid database, EMBL, GenBank, DDBJ. These are the example for a primary nucleic acid database, and for proteins, PA, protein information resource, MIPS, SysProt, TM, TREMBL, and NR3D. These are the example for protein database. Okay, apart from the sequence, literature, domain, structure, we can get data from NMR crystallography. So whatever the information you have derived from NMR and the X-ray crystallography, we can upload the information to databases, define the data structure in the database. The PDB database, protein data bank, is a suitable database for storing structural information. Or we can do the molecular modeling by which we can predict the structure after the prediction, we can upload the predicted structure in the database, PDB database. So docking is an important technique by which we can predict the ideal orientation of ligand in the active site of the receptor when bound to each other to form a stable complex. So the molecular docking plays an important role to find the function of the protein and the functional feature of the ligand. So it works based on the locking key model. There are two different types of docking. One is rigid docking and flexible docking. With and without the interaction, we can classify the docking into two. One is a bonded interaction, another one is non-bonded interaction. In the bonded interaction, we can see the bond created between the two molecules, whereas non-bonded interaction, we cannot see the bond, but the orientation looks like the interaction. Then the tools used for molecular docking, these are the offline tools used for molecular docking. X, Dock, Autodock, Vina, Pyrex. So these are the offline tools, and for online tools, Backstop, docking, server, system. These are the few examples for online tools for molecular docking. Okay, for molecular docking, you need a protein and a ligand. Both molecules must be prepared in a proper manner. Protein must be derived from PDB database, or if you have NMR on the X-ray crystallography, from that we can derive the protein structure or the receptor structure. And the ligand can be derived from a huge number of ways. One is from literature or the phytochemistry, GCMS. From GCMS we can get the structure, or whatever the way you have derived the structure of ligand, you can have it. But whatever the structure of ligand you are having, it must be properly converted into 3D structure. You must have 3D structure of receptor, that is protein, and the 3D structure of receptor. The 3D structure of receptor and the ligand are very, very important for molecular docking. Once you have ready with receptor and the ligand, it must be properly prepared. For receptor, enzyme inhibition must be used because the free electrons are properly managed. So generally you can do the docking properly. And for ligand, it must be validated properly whether the ligand can be used as a drug or not. Otherwise, it won't be based of your time and other resources. So the preparation of ligand and receptor is very, very important. For preparation of ligand and the receptor, there are huge number of softwares are available. And to validate the ligand, you can use the Lipinski rule. So where the 5 important parameters are validated. So once your ligand obeys the 5 parameter, then the ligand can be used as a drug molecule for docking process. So validation. After the completion of molecular docking, validation is very, very important. So for validation, we can use Ramsey method for validating your receptor, verify and the project. These are the validation tool to validate your protein. And to identify the active site, what if active site finder, back finder can be used for active site prediction. So here you can see the receptor structure, the right hand side and the left hand side, the receptor tightly binds with ligand. So this is a kind of tubulin protein. The tubulin protein binds with takes up as a ligand. The yellow color indicates the ligand very tightly bonds with a receptor, that is particularly at the Taxol domain. Because the Taxol derivatives is one of the most important drug molecule used for cancer therapy frequently. But from the Taxol domain, the tubulin has another two domain. One is GDP binding domain and the GDP binding domain. So here, this is the flowchart to carry out the molecular docking. the docking is confirmed, you can validate whether it is performed well or not. The docking can be validated by its score, docking score, binding energy, bond length, and the distance between the atom. Always the distance between the atom is expressed in Armstrong. So you can use these three score to interpret the result. Among which the bond length is very important. If the bond length of the docking has very minimum score, it means the docking is tightly performed. If the bond length is lengthier, the docking is performed in a loose manner. So shorter bond length infers the tight interaction, whereas longer bond length infers the long interaction. The tight interaction always the significant result in molecular docking. So here you can see how the bonds are created between the two atoms. The large one, the large structure, this is the structure of ligand, and this is the structure of receptor. So here you can see the receptor is tightly interacts with ligand when compared to the another. This is the example for protein-protein interaction. The protein-protein interaction viewed by another tool, Lickplot. The tool name is Lickplot. Three structure can be viewed by PyMol. So PyMol is a very good software to visualize the 3D structure of the protein, and Lickplot is a very good tool to view the two-dimensional quantum transformation. So this is the Lickplot view of molecular docking, and this is the 3D structure of molecular docking. So molecular docking is very important tool. So one can perform molecular docking to interpret your research for different purpose. And if you are from any field of biology, without any difficulty, without any doubt, you can use molecular docking to solve the problem.